Great. All right, welcome back, and here we are back in class, and start here. So this week, we're working on your capstone projects and papers. That's prominent now in your activity, I hope. And we're talking about learning about teratogens, and we had a great class discussion about them on Monday. And today, my goal is to talk about mechanisms of teratogens. And so we did the same kind of thing in the first half of our course is let's kind of get going with it, understand, some of the topics at the level of principles and concepts and terms, and then let's try to look into mechanisms. And it's the same idea, that is, there's been some disruption of a developmental process. Well, developmental biologists know a lot about process as that occur, and they've learned about them in a small number of model organisms, but it just turns out that you can just cross all kinds of evolutionary time and space with developmental mechanisms that are highly conserved, and many things we're talking about are common in many organisms like that. So we ought to do the same kind of thing in teratogens. And I've described, as we're introducing teratogens, that the teratogens a little bit frustrating. There's a little bit of a kind of a sort of a crowded, confusing thing where the teratogen could act on, or endocrine disruptor could act on, really any step, and maybe multiple steps in an organism's development at different times, in different ways. And so in trying to organize that into learning and learning about mechanisms, it's good to try to pick and choose, and pick examples like I described last time that maybe have primarily a single mechanism and a single effect that they have, or at least that we could focus on in order to get down into I see we're learning about pathways and understanding those as well. So we want to do that. And for Monday, our goal is for you to do some of that. And so we had a really good discussion. In preparing for today, I suggested on Monday maybe I could follow up on them, because we talked about hyperthermia and other topics, that were new or mostly new to me, and I was like, I want to learn more about it. And I just haven't had a chance to do too much of that, so maybe you will. Or maybe something will come up in class today that you'll pursue. And the idea is to bring that to class and bring examples that you think would be good to contribute to our overall learning from our course, because we want to have those be things that we could write about on our final exam. And so that's a goal for Monday, and as always, email me 5 things before class. If you want to bring and show a PowerPoint image or just the title of an article or a figure from an article, email those to me before class. It seems like I just don't have much chance to look at things very well before class. So if you come to class on Monday and you can just say, hey, I'm going to want to show this, just say that, and we can get it ready to go. Okay, so that's that. All right, then what I want to do is just read through, since you're working on your capstone, just read through what the syllabus says about the capstone, or part of what the syllabus says about it. Just, and I've talked about all this, there's nothing new, and it's on the syllabus, and it's just an overview of what you're up to, but just so you have a kind of reminder and nobody gets confused about where we're at with that. So I just want to read through this from the syllabus. About half the course, and that's true, about half your effort, half your grade is going to be based on that capstone project, and each student will have 2 Google Docs, and one is the term paper work or the journal, and the term paper writing. And I'll be able to look at the history to see, that there's a decrease in likelihood that you're using AI for writing. And I've emphasized throughout the course more and more, I think, wow, AI for research is fantastic, and I'll continue to do that today. I'll get examples today of helping find things and get, some help for AI. And just use that as a resource, list it in your journal, include it if it seems appropriate in the bibliography of your term paper, but not for the writing like that, okay? And so we've said that a bunch of times. You need to write the term paper, and one way to demonstrate that you're doing that is to work on the 2 documents as you do the work. Don't work and write in some other document and paste it into the 2. shared Google Docs. Instead, you could paste pictures of your notes or your writing to show that it wasn't, something that just came off the web and you pasted it in. I'll share it to Google Docs, and I can use track changes so I can see that like that, okay? So you can use AI resources to help you learn about specific topics, include that use of AI in your bibliography, but you need to write the capstone paper. One way to demonstrate that you're doing that is to work on the two documents as you think it planned. Don't work and write in some other document and then paste it into the two shared Google Documents. You can paste pictures, blah, 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 like that. There's no specific format for the capstone paper. We've talked about that. Cite your references in the paper, then include them in a bibliography. You can use any sources, just make sure to cite them and include them. And there's no format for the specific format, just so I or anybody else can find, what is that resource? And so they can be, again, information from other classes, other textbooks, other instructors, experts in the field, websites of any kind like that. So that's that. Okay, then here's the next thing. So in getting ready for the day, yep?
I think the due date for that is next Wednesday, am I mistaken?
So, yeah, so Luke says the due date for that is next Wednesday, so yeah. And so what's going on with that? So what's going on with that is that at the University of Southern Maine, the instructors cannot have any major assignments due the last week of class. So it's due before the last week of class, but I don't care. Like that, it just have to, by the rules of the university, put it in there as that's the due date, but you can finish it, and you could even finish it after finals week, like that. I always want to be careful for a student to not end up, they got it incomplete, no, they're going to finish over the summer, right? That's just not a good thing. But if you get jammed up like that, again, for official, thank you for bringing that up, for official purposes, it has to be then, like that, and that's why it's there. But yeah, and then there are kind of two personalities. One personality is a student that's like, there's a deadline. I can crank it out by the deadline because I'm going to stay up all night, stuff like that, or really prioritize it and get it done. And that's for them. It's good to have a strict deadline. Then there are other students that get stressed out by the deadline, and it's bad to have a strict deadline. And I have said that to millions of, well, not millions, but let's say millions of students, one-on-one. I'm like, okay, I don't know whether I should give you a strict deadline or say, let's just get it done, right? And so I say, let's just get it done, right? And we can communicate about it on a one-on-one basis, where you're at, and continuing to do that. And don't hesitate to do that. Don't be hesitating because you're like, you haven't gotten much done. Just communicate with me on how things are going, where you're at. If I can help, that'll be great. And again, it's absolutely fine to get it done, at your timing, but get it done. Yeah. So thanks for bringing that up. So, other questions people have? Read up.
Can we have like a ballpark of like the length?
Yeah, so that's a really good question, right? Students really always want to know that. And in my work, it's mostly lab work and lab notebook work. And in a lab notebook work, you want it to be complete and concise, right? So you don't have to add BS to reach it to a specific length limit, right? And also, when it is BS, whether it's on an essay exam or on like a lab notebook or a capstone project, it's funny because as a student, I thought I was really good at, just like, 30 pages, I got this. But as an instructor, I'm like, oh my gosh, it's just like blatant where the student tips into this thing about, I got to stretch this out a little bit, So there's that, right? That's my, spirit of my work is let's get it done, right? And so short is good, right? Like that. But then, I don't want people to shoot themselves in the foot. I think somewhere I say like 15 single-spaced pages. I think, or maybe I've said that in previous years, like that. But again, you know, it's to kind of underscore it. It's half your grade, you know, like that. That's saying about half your grade, the overall effort, the journal, the capstone, like that. And and I look at it all. But at the same time, you know, if you have some, you know, it's really good to just have some solid stuff, you know, and frame it. I think I mentioned this before. I like to mention it. People are pretty familiar with the idea, oh, they went to like a science talk and they left and they said, oh, that talk was a mile wide and an inch deep, right? And that just means they just covered it superficially, they didn't dive into it. Then nobody ever says the opposite, which is that talk was an inch wide and a mile deep, right? Where the person just got way into the nuts and bolts of it, right? And in any presentation, whether written or oral, it's good to have that balance, right? It's good to have a really good balance like that. So think of that too, right? Get a good overview and then dive into some feature of it, right? Like that. And in this course, in the past, students have usually cut off too big a picture, you know, and they're trying to carry it too much of it. So think about that, you know, getting a good overview, but then leave some of that behind and dive into a specific thing. One thing is, and I'll show you with this example, because I want students to get to the primary research literature. So we're going to talk about that some with this example to really get some science articles, right? Because we've been talking about the, you know, short writing assignments were very different than like a term paper. But it's really important to get into the science discovery and where scientists are at with the mechanisms. We have talked about mechanisms, you know, in our course, and we want to get to how scientists are finding the mechanisms. So I have that in this example. So that's a part of it too, is part of the getting, you know, diving into some aspect of this. And it doesn't have to be diving into all of the things, but, you know, get a good one. And then, you know, imagine that, you know, I think the last thing is that I always, you know, and you know, I think any instructor would be like, oh yeah, if the writing connects it to our course topics. And you know, you remember to write it as if you're writing to a classmate, and so the classmate wants to hear stuff that's reminding them of something that we've learned about, and you can even do the language in the thing where in class we learn, you know, in our course we learned about this, and it's a, you know, you're welcome to do different kind of writing styles than are typical of, you know, published science research language. That's fine, you know. You can use first person a little bit, stuff like that, that's fine. But think about that. Think about trying to bring up, you know, as you're introducing it and diving into it, what are some things we talked about in the course? You can compare the topic to other ones in the course. Both how is it similar, how is it different, how is this more challenging, like that, that's great. And then what are you teaching the students in our class that's newer, that's deeper, you know, more like that. That's good, that's really good. And you know, that's kind of sucking up, right, to the instructor, which I remember being in college and I was just like a star of that. Where I'm like, wow, is that a new shirt? Like that kind of stuff. I mean, you know, yeah. So I mean, I don't need any of that. I do not need any of that. But it's good to write that way, to connect it to the course, compare and contrast, connect it what's similar, what's different, like that. That's great. Yeah. And another thing is I don't have time to give feedback on draft writing. That can come up sometimes with students, and it's just not. When I read something, I have to provide. I'm just trained to provide massive feedback, and I just can't do it. We can talk about it and talk about where you're at, but as far as like getting and saying, "Am I all done?" I can't really get that done. Yeah, you know, we can talk about it like that better than just sending me something like that. Yep. Other questions people have about anything at all? So again, we're heading toward, on Monday, you finding molecular mechanisms. And remember this idea that in our course, the way we're going through our course and our activity in our course, we're actually modeling the capstone project work. like that. So I use this example to dive into some primary research literature, okay? So, and you can interrupt with any questions like that. But does anybody have anything else about that capstone or anything like that? Yeah, okay. Yeah, and again, thank you for bringing up that deadline. I spaced that out, but that's why it's there that way. Yep, all right. And so here's this, right, is that, you know, I was learning about some mechanisms, and we've talked about some metals. Juliet, I think, brought up lead. There's methyl mercury in our book. And when you look on the web, there are other metals that are important. And one of them is this arsenic. And we haven't talked about arsenic at all. And so what I did, how I got to it is I was just like, you know, metals are toxic, and people know that metals are toxic, like lead and so forth like that. But what about endocrine disruptors? And I'd like to go back to that, because when we think about endocrine disruptors, we have to always think about, well, is it going to be binding directly to the receptor and really messing up the receptor some way, or is it some way on the path, the synthesis of the path, or the cells in the endocrine gland that make it? So sometimes the cells in the endocrine gland that make it can be affected by this, you know, this factor. Or is it downstream of the signaling, like that? So things like that. And I thought, oh, you know, that would be a good thing to show the students that, you know, these metals could be endocrine disruptors, because, you know, there's so many things a metal could do that could be toxic and problematic, right? And so, you know, I just Googled, you know, this, you know, could metals be endocrine disruptors? And they're like, yes, right? And so when we think of things like lead, we don't think about that as its primary problem. But it turns out, yeah, it can be an endocrine disruptor, like that, okay? And so it says, you know, by affecting hormone synthesis, signaling, metabolism, right? It can affect any step in the pathway. So again, to kind of just underscore what we've talked about that. And then one of them that popped up that I hadn't, you know, included in our course is this arsenic. And it says arsenic can be an endocrine disruptor by affecting steroid hormone receptors. Now, that's not the primary mechanism of arsenic toxicity. Arsenic causes lots of different things. So we want to be careful about that when we say, oh, we're going to dive into arsenic a little bit. We're really not always diving into the primary concern. But one thing we also want to remember is when we dive into things that are endocrine disruptors, lots of times that's the problem that has the most sensitivity to the toxin. So that you know, we drop down, and lots of times it is that disrupting hormonal pathways where is often the most sensitive. And so sometimes there'll be chemicals and so forth, and environmental factors that are understood to cause problems, and you know, the government has set up a threshold concentration, and now they're redefining that threshold concentration and dropping it down further, like that. So I just did it this, you know, I did this, and it's in the slides, you know, like that. But then I was like, okay, so here's an example, because this popped up for me, and I was like, okay, well, how's that work? What's arsenic do? You know, here it says affects steroid hormone receptors, right? So some of the other ones are affects the hormones, you know, like that. But this one says affects steroid hormone receptors. So what's up with that? Okay, and I want to just dive into this as an example of like how you're getting ready for Monday, or how you're doing your capstone project, right? Like that. Okay. So first, here's Doug Curry, and Doug studies arsenic, like that. So that's the first thing, is this idea that, you know, we're not just going to jump right to research articles and stuff like that. There's the reality that we have an expert, you know, in the building, like that, and Doug works on that. And of course, I know something about Doug's research, but not very much. But you know, if you had Doug for a course, you know, you could connect that to that. If you run into him, right? You just kind of don't want to bother emailing him, but you could run into him and ask him about that. Get some resources, because he would know, he would have a lot to say. And then another thing about it that he would say right away is, oh, this is a big problem in Maine, because there are so many private wells, and the private wells are drilled, and a lot of times they're drilled through rock, And arsenic is just naturally occurring in rock, and it will leak out into the water. And because they're private wells, we have a big range in Maine of issues with arsenic. It's a real significant environmental disruptor of health and development in Maine, with some wells being dangerously high. And how would that private owner know, right? Like that, okay? Here it says chronic exposure is linked to cancers and lower IQ, right? So the big primary issues with arsenic is not steroid hormone receptors. We're going to dive into that because what is up with that? But it's others. And arsenic is one of these environmental factors that affects lots of things. Doug is a neurobiologist. He's especially interested in that low IQ in children, right? So it disrupts normal development of the brain, like that. All right, so I just want to show you this because we haven't talked about it. You've seen me use AI a lot to kind of go into this, but when I start wanting to look at, for science literature, I use this Google Scholar, and hopefully, some of you had exposed to it, or all of you had exposed to it some. Let me just talk about it a little bit. So see, I wrote in arsenic endocrine disruptor. Like I'm doing a Google search, but I'm restricting it to scientific research articles. And I can restrict the type of articles. And so I just want to mention that idea as we're working on our capstone projects. And there are primary research articles, and those are ones that are reporting new scientific discoveries. And you should definitely have some primary research articles in your capstone paper. And more is better. So dive into it, really get to look at some of the work that's going on. And then there are review articles. And review articles are really helpful too. A review article sometimes is reporting new scientific discoveries, but that's not its main purpose. Its main purpose is to catch the reader up on recent discoveries in the field. And so that's very helpful too. And so you can control that. You can also control the time you're looking at. So sometimes you'll get some papers that are a little bit old, and you'll be like, oh, I should try to find something more recently. We can look at older work, but like our textbook that we're not using directly, that's 10 years old. Well, there's more recent stuff, and we'll talk about that in a minute too. And so, I did this search, and I was like, oh, here's one. Arsenic is an endocrine disruptor, effects of arsenic on estrogen-mediated gene expression. So, we talked about that. And I'm like, there's their steroid hormone receptor. I was like, that would be great. I could bring that to class, right? But just like with AI and research searching, you got there really fast. It's good to kind of scroll down and look at some stuff. And I scrolled down just a little bit, and I found an article, and it's a primary research article, so it would be a great one if you had this as a topic in your capstone project, or if you wanted to bring it to Monday, right? And because look at this title, Arsenic as an Endocrine Disruptor. Arsenic disrupts retinoic acid receptor and thyroid hormone receptor mediated gene regulation and thyroid hormone mediated amphibian tail metamorphosis, right? So good stuff, because we know from the first half of our course, thyroid hormone is what regulates amphibian metamorphosis. So you gotta have its normal function, so these people look at it in the tails of these amphibians like that, right? And so it's a primary research article. That's good. They're going to be discovering new stuff. And then just in the background of it, they say, this same group, they say, we previously demonstrated that arsenic, a potent endocrine disruptor at low environmental relevant levels, altered steroid signaling at the level of receptor-mediated gene regulation for all 5 steroid receptors. So that caught my eye. And, I note that, I know this field pretty well, right? And you don't know your fields pretty well. But, so I'm, acknowledging that I could kind of breeze through this faster than you could and catch some of the things that are like, oh my goodness. So 5 steroid receptors, and those would be members of the nuclear receptor and, estrogen receptor, testosterone receptor, glucocorticoid receptor, vitamin D3 receptor, are great steroid hormone receptors, and they're all nuclear receptors. this thyroid hormone receptor and retinoic acid receptor, those are not steroid hormones. Their receptors are members of nuclear receptor superfamily, but different classes. And so what these people are really driving at is they're like going like, well, so we found, they probably started out finding that original thing of, oh, it looks like that arsenic could cause trouble for the estrogen receptor, because people really study the estrogen receptor a lot. And then they probably said, well, what about some other nuclear receptors, you know, like that? Because is it specific to estrogen? And they probably looked at the others, and now they're looking at more, and they're finding these other nuclear receptors are also affected. So it's intriguing, right? Because I started out on this path saying, I'm going to demonstrate to the class that these metals can be endocrine disruptors, even though they probably don't interact with the receptor. And here I'm finding, oh, they can interact with the receptor. Now it's a metal, right? So could it possibly be a mimic that binds to the ligand binding domain and mimics the estrogen hormone? How could it be that, right? Okay, so what's going on, right? I thought it was really interesting to dive into this. And I just want to show you this, that that article was, 10 years old, like that, So if I ran into that in class, in my capstone, right, I'd say, okay, I'm going to look into this, but there's going to be more recent stuff. So, what's their take-home message? And Google Scholar has this feature where when you have an article that you're interested in, and you have it in Google Scholar, it shows how many times and who has cited it. So it's a way to update what you're looking at and what you're learning about. And that's just that's my favorite feature of Google Scholar, is that ability to move up to current time by finding more recent articles. You can do this by just asking for more recent ones, but adding this to it and then maybe adding that date to it also really helps you bring it up to speed. So Google Scholar has those features that are really useful even in the day and age of AI. Google Scholar, I think, is still very good. All right? Yes, okay? So back to just the article. In the conclusion of their article, they say that arsenic had similar effects on retinoic acid receptor and thyroid hormone receptor mediated gene regulation as those previously observed for the steroid receptors, suggesting a common mechanism of action, okay? And so when I saw that, I was like, well, what is the mechanism of action? This is 10 years ago, right? What do people know? So I could have done this, right? I could have tried to look for people that cite more recently, but a lot of people have cited it, and who cited it about that common mechanism of action. So that's where I think combination of doing Google Scholar type work in science and then going back to AI can be really helpful, because I just typed in for AI common mechanism by which arsenic affects several nuclear receptors, right? And AI just grinds through it fast, and it says this, and it's just like, oh my gosh. For somebody who's interested in these topics, it says, it explains it. Primary common mechanism by which arsenic affects several nuclear receptors is through the direct binding of trivalent arsenic to sulfhydryl groups in the amino acids in nuclear receptors. So it is binding directly to the receptor, but not by mimicking the hormone and binding to the hormone binding domain, but rather binding to some of the specific amino acids, the sulfhydryls, the cysteines, and inhibiting binding to the DNA. Okay? So it says binding thiol groups. Arsenic particularly is high affinity for sulfhydryl groups, disrupting the structure of zinc finger motifs within the DNA binding domains of nuclear receptors. And again, I know about this topic, and I'm like, whoa, that is interesting. So I just want to show you that. So that these nuclear receptors are all transcription factors. They're called nuclear receptors because they function in the nucleus, and they bind to DNA to be transcription factors. And they bind to DNA with a DNA binding domain that directly binds to the DNA. I just have an image of that. So there's DNA, and this is the DNA binding domain of any of the nuclear receptors. They all look the same, and they all have a very small region of the big nuclear receptor that binds to the DNA, and this is just showing that in blue. And you might be able to see that it's binding, like in the twisted DNA. The twisted DNA has these grooves, both the what's called the major and minor groove, and it's like the just short parts of the nuclear receptors are sliding into that groove, and they're held in a very specific conformation because the DNA binding domain of every nuclear receptor includes zinc atoms, and the zinc atoms are held or coordinated through non-covalent bonds and holding the protein in this portion in a very specific conformation like that. And so scientists call this organization a zinc finger because it's coordinated by, held by zinc, that's essential, and then it looks like the protein has got like fingers that are sliding into these grooves. And so the structure is that here's the primary amino acid sequence of a zinc finger. There are the fingers, and then see the role of zinc in coordinating and holding that together. And how is the zinc being held? By cysteines, by sulfhydryl groups coordinating it. And that's what arsenic does, is it has a higher affinity for those sulfhydryl groups than zinc, and it just destroys the ability to bind to DNA like that. And so I think for us, I got to it quickly because I know about it, but just for all of us, I think, you're like, dang, that's a way, to get down to the molecular mechanism, right? Because you say, we're going to be an endocrine disruptor. It's going to disrupt these, and it's going to disrupt any nuclear receptor. Because all the nuclear receptors are one gene family. I have an image of that too. Here's just an image of just the structure of the coding region of any nuclear receptor, where it's organized like this, where it's got an activation domain that's going to cause changes in transcription, DNA binding domain, that's a zinc finger binding domain, and then a ligand binding domain, right? And so they're all conserved, they're all similar. And if you have something that causes this problem, it's just really a good example where, oh yeah, it's an endocrine disruptor, and it's just across the board. Now, would it be an endocrine receptor for another receptor that's not a nuclear receptor? Maybe, but not necessarily. On the other hand, arsenic is probably a problem for any nuclear receptor. Whew, like that? And that level of arsenic might be lower than some of the other problems that arsenic causes, and may be sometimes more subtle than some of the problems the other arsenic causes. So I thought that was just a great example to talk about like that. Then again, I understand it, but here's back to that, it was a primary research article, and they have methods, right? And the methods they used are that they actually included, let's see. Yeah, so I just got confused myself a little bit, right? Because this is that article that was talking about amphibian metamorphosis like that. So they're working on that, but for testing it out, right, they'd already done this testing work on other nuclear receptors, and so they use the same strategy. And maybe you'll get down to this level with your capstone paper, and that's great. Just one time, maybe you'll get down to it and be like, look at this, pretty interesting. I was, happy to read this primary research article. So I want to show you that, but maybe you won't, quite get down to a place where you really want to write about, the methods from a paper. So, because if you just said, I'm going right to the methods, that would be like, that's going to be a mile deep, right, and an inch wide, right? We're really kind of diving into this example. But maybe you find an example where it is worthwhile to dive in that far and explain it some, So I'm ready for either. But in this example, it's worth it talking about, because what they're doing here is all the nuclear receptors are transcription factors. And you can assay the activity of a nuclear receptor, any of them, by assaying gene expression. And so what this lab did, remember this is a lab that said, oh, we already studied it on all the steroid hormone receptors of the nuclear receptors of the family, and now we're going on to thyroid hormone receptor and retinal calcium. How are they doing that? So, sometimes it's satisfying to get to that. Because what they have is, even though they're really studying amphibian tails in this paper, they have an assay system, and it's just a cell-based assay system, and it doesn't even involve amphibians. It involves human cells and rat cells, and they're growing them in a dish, and what they can do is they have a gene that they use to test the activity of any of these nuclear receptors. And the gene is designed where they put in a binding site for the receptor they're studying, and then the transcription unit, the expression, is going to be a gene that's the gene that causes fireflies to flash. So it's called luciferase, and that protein can produce, in its enzymatic reaction, it can produce light. And so they have a light-based assay for testing how well the receptors work. And it's in a dish in a lab, and they could just add, they just set it up to have the appropriate binding site for whatever nuclear receptor they want to study. So in this paper, binding site for a thyroid hormone receptor, and they ask, well, can the thyroid hormone receptor work? And they add some thyroid hormone receptors, and the cells light up in a dish. They have a assay for detecting luciferase. This luciferase assay is very, very common, like done in many, many, many labs. So you can do that, and then they can just say, oh, yep, we can activate the luciferase gene with the thyroid hormone, and then they just started adding arsenic, and they're like, whoa, there it goes, you disrupted it like that. So really cute assay, and if you run into that and you can write about that, great, right? If you get close to that, great, because you do have to get into some primary research literature, and sometimes, if you showed me what you're looking at, I could help you. A lot of this stuff, I don't know, that's not my field. But in some cases, I'm like, oh yeah, you should check this out, because that method would be a good thing to write about, like that. Yeah, okay? So I just wanted to dive into that example a little bit, both for, Mondays. And again, one last time, you're not expected to just be able to crank down into it like that. Mostly you'd be like, this is just curious, like that. But, you can run into something where you might have some other knowledge, in some other, course or, experiences you had, that you can match up and be like, this is like, can explain this. And so whether it's bringing something on Monday or in your capstone project, I just wanted to give you that example, both, how to use Google Scholar, and also that idea, combinations of Google Scholar and AI overuse and stuff like that. Great, okay. So that's that. I should stop and see if anybody has any questions about that. I just pulled that up as an example to kind of model for, what we're doing in our course. All right? So here's the next thing, right? I've talked about how, we got to talk about, try to talk about the, what's the pathway that's disrupted. And so we did that in the first half of our course and tried to dive into pathways, and that's what we're trying to do now. And so I have mentioned a little briefly that all of this fits into this big field of toxicology, which is a field that's not limited to biology. It just covers a lot of territory, because it involves governmental regulations and so forth like that, And so I looked a little bit at some toxicology stuff to try to figure out, like, well, how would what would a toxicologist add to this? And so here's this, and you'll see where we get to with this, okay? So in modern toxicology, and I talked to, we have emphasized that traditional toxicology really stumbles on some of these modern topics, like an endocrine disruptor, and they're just like, sensitive period, that's tough, like that. I mean, they are aware of it, but it's a tough thing, and Luke brought up, the non-linear dose response curves, that's really hard. So modern toxicologists like to do something they call an adverse outcome pathway. And we're just using that to help frame our own work and the way we do it, you know, as biologists, right? So it's used in toxicology as a way to systematize our understanding of toxins. And remember, there are these really three categories of toxins. So one's acute toxicity, that's where, you know, the organism just falls down and is in seizures and dies. That's, you know, happening now. And it doesn't have to be lethal, but it happens immediately. I think the definition is within 2 weeks that I found. And the other is delayed. That's the one we're more likely to be focused on in our course. Oh, we don't see it for a while, in, you know, some cases years, right? And then the other toxicity is chronic toxicity. That would be like smoking or like arsenic in a well water where you're drinking it every day. So just understanding of toxins. And so these AOPs are used in toxicology in risk assessment and regulatory applications. So really, you know, something different. It's a different field. But how can we use that logic to help us with our course as biologists? And so here's what they do is, it'll be a document they produce, and they'll produce it and they'll say, what's the evidence for the molecular initiating event? And what that means is, how does this toxin, you know, what is it binding to, or what is it directly doing? So for the thing we just talked about, the molecular initiating event is, well, the arsenic binds to the zinc finger domain and disrupts the zinc binding to it, and you know, blocks its ability to function as a transcript effect. And we're just going to jump from that down to 3. What problem does that cause? Like that, right? And then in between are the steps that are something we really like to focus on in our course. What's the mechanism? What's the pathway? Is it going to affect gene expression? Is it going to affect DNA methylation? Is it going to affect hormone action? You know, what are the steps like that? And we don't need to remember their keyword, you know, their terms, but it's a nice thing because we've been talking about it. And you can see toxicologists are talking about it too. And this is how they want to organize it into a 3-step thing. You know, what's the first molecular event? What's the pathway? And what's the ultimate, you know, effect? And in our case, we wouldn't say adverse outcome. We'd say phenotype. What's the phenotype? Like that. Okay. Okay. Just to show you that, and then to just talk. Show you this because we talked a little bit about this mind mapping or concept mapping. It's a mind mapping thing, right? They're also going like, well, it's a timeline, right? You got the initiating an event, you got the outcome, you got key events in between. So they're organizing it like that. They're organizing it like that. And then they produce these things where they'll start to try to, like they'll say, well, if you have multiple toxins and they have a common initiating event, we'll put them in the same chapter of our topics, like that. And so it's good to think about that, about how we're organizing these and comparing these. Like the first slide I showed, I said, I was going to put all the metals together. That's not the initiating event. That was just the category of the toxin, metals. You know, that's you know, kind of arbitrary and not what a toxicologist would do. Or a modern toxicologist. Okay, so here's a good example of that, right? We said before, oh, this is a pretty good example because we know the steps, and there's a primary story here, and it drives down from that to the key events to the adverse outcome, right, like that, and we understand the molecular pathway pretty good. So it's a good example to think about this linear pathway. And that arsenic one is a good one I just talked about too, right? So we have some good examples of that, and those are good ones both to try to get organized to bring to our final exam, to be ready to share, like that. And then also for your capstone project to try to think about, okay, what is the way to organize your capstone paper? So I think this is helpful to see that timeline. It's pretty, I think it's pretty obvious, but it's nice to see this big field of toxicology agrees that that's the way to fundamentally organize this. So DES is a good example of this pathway story. And I mentioned before that some of our examples are good and some of our, like fetal alcohol syndrome has just so many impacts that it's hard to use as an example, even if it's a profoundly significant one. So here's just some more things that I got out of looking at those toxicology topics. So the steps are like this, you know, what's the cause of the disruption, the source, and the mechanism, and then and you can reflect back on the example we just covered, the arsenic and the nuclear receptor family. Then biological processes disrupted, gene expression changes, and the effect on the phenotype like that. That's what they're doing for that. Pretty straightforward, but very good to see that. And then just a little bit more on those three steps, the source and the mechanism, right? So we want to be aware of that. You know, what is the thing and what is the mechanism that the thing does, you know, direct thing. It started out where I'm like, oh, I don't think a metal would be an endocrine disruptor by binding to the receptor. But then I found an example where, yeah, that is its mechanism, like that. And then we haven't really talked about this, but I think it's good to organize our learning based on what is the nature of this factor, the toxin, the teratogen or endocrine disruptor. So we have some good examples of drugs. Because like, remember on the first exam, I said, oh, we're talking about phenotypic plasticity, and I said, give me an example of one that's this, and one that's that, and one's that, right? We had like 5 different categories of thing. And so I think for learning, I think it's natural to also organize some based on that. You know, did we talk about some examples that are drugs? Yes, and that's new, right, to a student. So you know, a student can write about that. Drugs are part of the environment, you know, and it's, you know, oh, as soon as you say that, yeah, oh yeah, they definitely are. But we don't think of it, you know, we don't think of those as being environmental factors like that. Or is it part of, oh, and so this thing, I don't know if you've heard the word built environment, but built environment means the stuff that humans make and use. So BPA would be built environment stuff. It's not a drug, it's not a pollutant, it's something that humans make and use. One of the things that I don't think we've talked about at all is, the book talks about some are flame retardants. There are a lot of chemicals that are put into the built environment for a variety of reasons. But BPA would be one, the phthalates, that's the story of that, you know, putting into plastics. And just based on PCBs are built environment things. So things that weren't intended to be like a pollutant, like a discarded, you know, thing that was waste material, but just part of our natural environment that was not nature, right? Or was it a pollutant? So that means something that was just released into the environment as waste material, or the natural environment. And so, you know, we can find examples of each of those, and I think it's good to be aware of that because it helps people understand, oh, these are all parts of the environment. So trying to think of a couple of examples from each of that category is a good one. And then with regard to that source and mechanism, I'll, you know, try not to mention this again, but, you know, is there primarily one story of this that's causing a problem, or is it lots? And I think that's a good one for us to distinguish between in our course, just for the learning purposes. You know, it's a course, we're trying to learn the things, trying to hang on to the ideas and principles, and by picking examples that have, you know, primarily a good and kind of maybe well characterized medical mechanism, I think that's great. Okay? So then these key events like that, the steps involved in, you know, in the organism for the problem, it's good to appreciate that, right? This is a toxin and it binds, it does something, you know, initially like that, but then what's the pathway and what's the steps? So we've talked about sensitive organisms, period. We've talked about model organisms, not so much in this part of our course as the first part, but they're relevant here too to help us understand, well, what could the key events be, you know? So with regard to, you know, hormonal disruption of sexual differentiation, what could those steps be? And model organisms are really great to open the door to understanding what could the pathways be. And then we've talked lots about the, as a step along the way, it's pretty common in development for it to include changing gene expression. Development almost always, involves, well, the cells have got all the genes, they're going to change them. And so we have that term epigenesis that both means changing any gene expression and also is focused sometimes on DNA methylation. And then the last thing I wanted to mention is this idea of phenocopy. And that just means that some of these teratogens or endocrine disruptors are creating a phenotype that actually is very similar looking to a mutant phenotype that could be identified as well. And lots of times those can go hand in hand, where you're like, we see this phenotype. Is it like any phenotype you've ever seen before? And you're like, yeah, we have mutants in lab in zebrafish that have this same phenotype. And so that can be really helpful. And there would be more to add about the pathway, and that's that mechanism pathway, like that. And then the final thing, the phenotype, well, is it an endocrine disruptor we're talking about, a teratogen, or both? And remember that, we have definitions of those. Here's something we haven't mentioned before, and that is the effect on the phenotype something you observe by studying the structure of the organism, so you can see the changed phenotype under a microscope or some direct measure of it? Or is it a function, like an IQ test, right? So yeah, with the lead, you see a change in the kid's brain by looking at it under a microscope, or do you see a change in the kid's brain by them doing a task? And that's something just to be mindful of those two. It's going to be, those two are both aspects of phenotype, like that. So that's that, and so that's that. And so just kind of trying to help us, guide us toward those goals. And then I have just going on to some different pieces of kind of trying to frame some of these. So another thing I did is I said, in those categories of, well, is it a drug? Is it a pollutant? Is it built environment type thing? I just did this. I said, well, let's look at the molecular mechanisms of pollutants that are teratogens. So I guess I kind of did a typo there. But just say, well, which of these, what are the famous pollutants that are teratogens, like that? And which of them do we have a good understanding of what their mechanism are, like that? And so some of them are endocrine disruptors, and back to that arsenic one, well, is it the receptor, the ligand, or is it something on the pathway that is the problem? Is it a problem in gene expression? Is it a problem binding to other molecules? So inhibiting an enzyme or oxidative stress. And so, what is we can talk about, biochemical pathways, but we can also talk about pathways using these kind of words, like that, right? Here's just the thing, we haven't mentioned oxidative stress. That actually is a very common part of the key events or the steps in teratogenicity or endocrine disruption. And so I put a little definition of this up here. So oxidative stress is an imbalance in the production of free radicals plus peroxides and their removal, and also repair of DNA damage they cause. So our bodies are always metabolizing nutrients, and those nutrients, metabolism of nutrients and other cellular processes produce free radicals. And those free radicals have to get dealt with, and so both by trying to remove the free radicals or inhibit them, and then also repairing damage that they cause. And so oxidative stress is one key event that leads, from the pathway of what got this started, and then on the way to how it's a phenotype going to be affected. Oxidative stress is a common mechanism along that pathway of the events that take place. So then I just started trying to look at and try to, pull together little summaries using AI of, some of the ones we're talking about, because that's a good thing to do too, is to organize your learning based on, well, we talked some about BPA. And so what about the mechanism, you know, like that? Because we learned some about, you know, like the kind of the nature walk type approach, you know, we learned, oh, BPA, it's in plastics, and it can be an endocrine disruptor, and so what about it? And so it's an endocrine disruptor, and it binds to estrogen receptors, like that. And then it has an effect, like that. So I made, I just wanted to show you that I started just kind of grinding out examples using, you know, that AI help. And I just have like several of these, right? Because I don't know how much you've caught, but we mentioned phthalates a little bit. Our book talks about phthalates, especially in endocrine disruption reading, a good amount. So can you get a summary of that? And what would the summary be is, well, yeah, you know, phthalates are in plastics, but what do they do? What's their mechanism of action? And so that would be this key event, the pathway stuff that is happening. And see, so here it comes up. It says, oh, they cause a lot of oxidative stress, like that. And so phthalates initially were studied because they can cause disruption in testosterone function. So they seem to be, they're both, BPA and phthalates are both in plastic, and they don't need to be there. They're just added to plastic to give the plastics a different performance quality characteristic. And the kind of traditional view is, oh, you know, well, that BPA affects the estrogen receptor, and oh, those phthalates affect the testosterone receptor. But now people are finding more and more of, well, it's this oxidative stress that's a big problem with phthalates, like that, okay? And retinoic acid, that popped up in our course, and so we talked about that. So what about a summary of that? And it's going to affect limb development. That's the one we kind of got going on in learning about it. And but it has other effects on the phenotype with that. Here's PCBs, you know, and I'm just rolling through these, you know, we'll post this recording, but also, you know, just to give you a feel for what I'm kind of trying to model, how you should try to, or how you can try to organize your learning for the final, to be able to say, here's, like, give me 5 examples, and you're like this, and this is what it does, and this is what it does, like that. So PCBs, okay? And with, I think, the time left, yeah, so we have 15 minutes left. I just want to show you this. So PCBs are they're part of the built environment, and they they're not used in like direct products, but they're used in some like oils and like in those big transformers that are hanging on the telephone poles outside. They're traditionally a lot of PCBs in them. And here, when talking about PCBs, they're an example where there's lots of problems that can be caused, but there's especially a problem with something called the aryl hydrocarbon receptor. So the PCBs become a good example for us to want to dive into mechanism and well, what is this aryl hydrocarbon receptor, and what do people know about it, and what that would cause for effects on the phenotype. So on that one, I wanted to dive in a little more because we haven't really talked hardly much about that. And so PCBs were banned in, I read it, like 1979. But they're just still present in the environment. I Googled it, I don't know if they had it on there right now. People talk about forever chemicals, and there are some chemicals that they have on that list. And PCBs are ones that people aren't thinking about on that list because the ones on the list are also, the forever chemicals are part of the built environment. They're in non-stick kind of cookware and stuff like that. But these PCBs would be falling into that category of like forever chemicals. The problem is they're just out there initially as part of the built environment. Now I guess would be a pollutant, left over and just not going away. And so another one that we should talk about some in our course in this category is there are some pollutants called dioxin, and they also bind to this aryl hydrocarbon receptor. And dioxins, I think, are like usually a pollutant that's derived from burning stuff, like burning PCBs, but also there's a little bit of dioxin, I think, made in just regular cooking, just a little bit, and not like when people used to, like for example, me up till third grade, one of my chores was to go out weekly and burn our trash. And I just stood in the smoke playing air guitar, right? And, you know, because it was school fire, and it was burning like styrofoam and magazines and trash, because there was no such thing as trash pickup. And if you wanted to, you could take it to the dump, but only people went to the dump. So dioxins are produced like a lot there as kind of like burnt, like high temperature derivatives of other built environment type things. Okay, and so lots of the PCB thing that people talk about include a concern about, oh, and are you talking about the PCBs or this dioxin derivative like that? And so just to show you a little bit, because we haven't talked about dioxin before, and so here's a PCB and a variety of molecules, and then dioxin is just really simple molecule like that. And both of them bind to this aryl hydrocarbon receptor, and so we're getting into that. So the mechanism of dioxin and PCB can be varied, but the aryl hydrocarbon receptor is a transcription factor, and it has a ligand that binds to it, and it's really interesting to talk some about this receptor. It's not a nuclear receptor, but it is another transcription factor that's activated by binding to a ligand. And the PCB and dioxins do several things, but they do bind to this aryl hydrocarbon receptor and disrupt its activity like that. And that aryl hydrocarbon receptor is a really curious and interesting receptor. It sometimes functions like a hormone receptor, so these are considered endocrine disruptors because they're affecting this aryl hydrocarbon receptor. So it's a really interesting because if you look at the ligand binding domain for the aryl hydrocarbon receptor, the binding domain is really big. And it was originally understood to be involved in sensing foreign molecules in our body. And those are called xenobiotics. So xeno means foreign. And so, for example, some medicines can bind to the aryl hydrocarbon receptor, and that's how those medicines are going to get eliminated from the body, like that. And the way that happens is the aryl hydrocarbon receptor is a transcription factor that can activate or increase expression of enzymes and a variety of tissues that detoxify molecules and foreign molecules like that. And so that's why it's called the hydrocarbon receptor. But then more recently, it's found that it actually plays a role in addition to with a specific ligands that are, so that's why it's considered more of a hormone receptor now. But it's a really interesting one because its normal function is to kind of help detoxify the body from foreign molecules. And dioxin and PCB are foreign molecules, and they bind to it, but instead of functioning so that it can activate gene expression and help eliminate them, PCB and dioxin block its function and prevent detoxification of those and removal of those, like that. Yep. And so here's just a picture of that happening. So there's a variety of different molecules that can bind to it and then activate it, and it activates expression, again, especially of genes that code for proteins that are enzymes that can help break down molecules like that. So it's a very interesting receptor, again, one we didn't run into. Oh, woo, yeah. And I think we probably aren't going to be, well, yeah, we can talk a little bit more. So in talking about how the aryl hydrocarbon receptor works, just to talk a little bit more about it, the people talk about a set of genes that inactivates, and there's a common set of genes that inactivates, and it's a whole bunch of genes that code for enzymes that are involved in metabolism, and especially in metabolism that would be breaking molecules down. And that includes molecules that could be a drug or pollutant that's gotten into the body, and the body considers it a foreign molecule, a xenobiotic, potentially a toxin, and it wants to break it down. And so aryl hydrocarbons have been a big story in toxicology for years. And here's an example where just unfortunately a couple of pollutants will bind to it and block its function, so it doesn't activate genes to try to get those expressed and help perhaps destroy those guys. Then in addition to that, that aryl hydrocarbon receptor does other functions in the body, and one of the functions that's prominent is immune system regulation. So yeah, when PCB and dioxin bind to the aryl hydrocarbon receptor, there are going to be issues with metabolism of foreign molecules, but there are also going to be issues of problems, phenotype problems with immune system function. So the immune system function is going to be decreased because we're failing to regulate genes that are involved in immune system function. So there are more of that, but I think I just limit it to that, we think about the aryl hydrocarbon receptor primarily playing this role in helping eliminate xenobiotic molecules, but it also plays other roles, for example, in the immune system. So dioxin and PCB will decrease the immune system. Dioxin and PCB are considered carcinogens. And I think it's not because they cause mutations. I think it's because they inhibit immune system function, like that. Yep. And then a little bit about this PCBs as these forever chemicals, and a deal with we've talked about before is issue with forever chemicals is not just that they are retained in the environment, but they bioaccumulate. So organisms are not going to get rid of them. And so it's just some of the forever chemical stuff, and the fact that PCBs and dioxin would be considered in this category, even though they don't pop up that much when people think forever chemicals these days. But in general, these forever chemicals bring up, and we talked about this a little bit. And I think that's probably, I have more slides, but let's leave on this one, right? So the classic modern forever chemicals are teratogens. So we should learn something. Well, what's the problem with them? They're in the news a lot. And our book definitely doesn't cover them. Those readings are 10 years old like that. And so just that idea too, right, is to bring it up, and maybe somebody will want to dive into the mechanism for Monday about one of these forever chemicals, and well, what is the problem that they cause, right? And we have that thing back from the adverse outcomes and the toxicology stuff is, well, what's the adverse outcome? And then what do we know about the molecular mechanism? What's the initiating event? And then what are the key events like that? So this is a good place to stop, and we'll chase down some stuff that you're interested in talking about next Monday. Anybody have any questions or issues with that? All right, I think that's a good stopping point, and we'll roll on. So we covered a bunch of stuff today, and the recording worked.